Block 2 Module 1 Case 1. I don't exist without you. Structural organization of the human body. The main idea of this case is that life is organized in layers. A human being may appear to be one complete organism, but that organism exists only because many smaller components depend on one another. Atoms combine to form molecules. Molecules contribute to cells. Similar cells form tissues. Tissues form organs. Organs cooperate in organ systems, and all organ systems together make one living organism. This is why the title, I Don't Exist Without You, fits the lesson. No higher level of organization can exist without the levels beneath it. The smallest level discussed is the chemical level. An atom is the smallest unit of an element that retains that element's chemical identity. Atoms interact through chemical bonds to form molecules. A molecule is therefore made of two or more atoms chemically joined together, and is the smallest particle of a substance that retains that substance's chemical properties. For example, 2 hydrogen atoms and 1 oxygen atom form a molecule of water, or H2O. Living organisms contain many molecules, but the major classes of biomolecules are carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates are important sources and stores of energy. Lipids form biological membranes, store energy, and participate in signaling. Proteins perform structural, enzymatic, transport, receptor, and contractile functions. Nucleic acids, particularly DNA and RNA, store and express genetic information. These molecules do not function independently. Their interactions create the chemical foundation necessary for life. The next level is the cellular level. A cell is the basic structural and functional unit of life. An organelle, meanwhile, is a specialized structure inside a cell that performs one or more specific functions. The nucleus stores genetic information, mitochondria generate most cellular ATP, and lysosomes participate in intracellular digestion. Molecules form the structures of these organelles, while the organelles cooperate to keep the cell alive. The tissue level comes next. A tissue is a group of cells, together with their extracellular products, that share a similar structure and perform related functions. The 4 fundamental tissue types are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue covers surfaces, lines cavities, and forms glands. Connective tissue supports, binds, protects, and transports. Muscle tissue produces force through contraction. Nervous tissue receives, processes, and transmits information. More than one tissue type then combines to form an organ. An organ is a recognizable structure composed of multiple tissue types working together to perform specific tasks. The heart, for example, is not made only of cardiac muscle. It also contains connective tissue that supports the organ, epithelial tissue, such as the endothelium lining its vessels and chambers, and nervous tissue that helps regulate its activity. Organs with related functions cooperate at the organ system level. The cardiovascular system, for example, contains the heart and blood vessels. These organs must coordinate to deliver oxygen and nutrients and remove metabolic waste. The human body is traditionally divided into 11 organ systems. The integumentary, skeletal, muscular, nervous, endocrine, cardiovascular, lymphatic or immune, respiratory, digestive, urinary, and reproductive systems. Although the endocrine and digestive systems were not shown clearly in the notes list, they are part of the standard 11-system classification. Finally, all organ systems function together at the organism level. This is the largest organizational level, the complete living individual. A failure at a lower level may propagate upward. A molecular abnormality can damage an organelle, injure a cell, disrupt a tissue, impair an organ, and eventually affect the entire organism. The notes also introduce the traditional 5-kingdom system: Monera, Protista, Fungi, Plantae, and Animalia. In that older system, Monera included prokaryotes, Protista included primarily unicellular eukaryotes, Fungi included organisms such as molds and mushrooms, Plantae included plants, and Animalia included animals. This classification remains educationally useful, but it is no longer the preferred modern framework. Modern biology more commonly recognizes the three domains: bacteria, archaea, and eukarya. Another important correction is that bacterial cell walls generally contain peptidoglycan, whereas archaeal cell walls do not contain true peptidoglycan. Cell theory, and the two major categories of cells. Cell theory provides the conceptual foundation for cellular biology. Its three classic principles state that all living organisms are made of one or more cells. The cell is the basic unit of life, and new cells arise from pre-existing cells. The final principle is particularly important because cells do not spontaneously appear. They are produced through the division of existing cells. Despite their enormous diversity, all true cells share several essential features. Every cell has a plasma membrane separating its internal contents from the environment, cytoplasm containing the machinery for cellular reactions, genetic material in the form of DNA, and ribosomes for protein synthesis. Cells can be divided broadly into prokaryotic and eukaryotic cells. A prokaryotic cell lacks a membrane-bound nucleus. Its DNA is usually found in a nucleoid region rather than inside a nuclear envelope. Most bacteria possess a single circular chromosome, although additional small DNA molecules called plasmids may also be present. Prokaryotes usually divide through binary fission. They are generally small, commonly around 0.55 micrometers, and lack most membrane-bound organelles. A eukaryotic cell has a well-defined nucleus surrounded by a double nuclear membrane. Its DNA is organized into multiple linear chromosomes associated with histone proteins. Eukaryotic cells also contain membrane-bound organelles, allowing different chemical reactions to be separated into specialized compartments. Most eukaryotic cells are approximately 10^100 micrometers in size. Somatic eukaryotic cells divide through mitosis, while germ cells undergo meiosis to form gametes. Prokaryotes commonly reproduce asexually through binary fission. Although they can exchange genetic material by horizontal gene transfer through processes such as conjugation, transformation, and transduction. Eukaryotic organisms may reproduce sexually, asexually, or by both mechanisms depending on the organism. The notes state that prokaryotic membranes lack sterols. This is generally true for bacteria, but there are exceptions, such as mycoplasma, which acquires cholesterol from its environment. Archaeal membranes are also chemically distinct because they contain ether-linked lipids rather than the ester-linked phospholipids typical of bacteria and eukaryotes. Protoplasm and the basic substances of the cell. Historically, the term protoplasm referred collectively to the living material composing a cell. It was once imagined as a relatively uniform, life-giving substance. Modern cellular biology has shown that the cell is not a homogeneous mass. It is an extraordinarily organized system containing membranes, organelles, proteins, nucleic acids, lipids, ions, carbohydrates, and other molecules. The term protoplasm therefore remains mainly of historical and educational importance. The notes divide cellular material into 5 broad substances: water, electrolytes, proteins, lipids, and carbohydrates. Water is the principal fluid medium of most cells. It accounts for approximately 70–85% of the mass of many cells, although cells specialized for lipid storage contain a lower percentage. Water is essential because many cellular chemicals dissolve in it, while other materials remain suspended as particles. Cellular chemical reactions occur among dissolved molecules, along membrane surfaces, or on suspended structures. Water therefore provides the environment in which most cellular metabolism takes place. Electrolytes are dissolved substances that separate into electrically charged ions. Important intracellular and extracellular ions include potassium, sodium, magnesium, calcium, chloride, phosphate, sulfate, and bicarbonate. These ions provide inorganic components for cellular reactions and are necessary for many control mechanisms. Sodium and potassium gradients, for example, help establish the resting membrane potential. Changes in membrane permeability to these ions generate electrochemical impulses in nerves and muscles. Calcium acts as an intracellular signal and initiates processes such as muscle contraction and secretion. Bicarbonate contributes to acid-base balance. Proteins are generally the second most abundant cellular substance after water, accounting for roughly 10–20% of the mass of many cells. Their concentration can be higher in cells specialized for contraction or protein secretion. The notes divide proteins into structural and functional proteins. CHON is sometimes used informally because proteins contain carbon, hydrogen, oxygen, and nitrogen, but the correct scientific term is simply protein. Structural proteins tend to form elongated fibers or filaments. Inside the cell, proteins such as actin, tubulin, and intermediate filament proteins form the cytoskeleton. The cytoskeleton helps maintain cellular shape, positions organelles, assists intracellular movement, and forms structures such as cilia, axons, and the mitotic spindle. Outside cells, fibrous proteins such as collagen and elastin provide strength and elasticity to connective tissues, tendons, ligaments, and blood vessel walls. Functional proteins are often globular and include enzymes, receptors, channels, transporters, antibodies, and signaling molecules. Enzymes accelerate chemical reactions without being consumed. During glucose metabolism, for instance, a series of enzymes breaks down glucose and transfers its chemical energy into ATP. Functional proteins may move freely in the cytosol or remain attached to intracellular membranes, where their locations allow reactions to occur in an organized sequence. Lipids are substances that dissolve more readily in nonpolar or fat solvents than in water. Phospholipids and cholesterol are especially important because they form the cell membrane and the membranes of intracellular organelles. Lipids constitute approximately 2% of the mass of many ordinary cells, although this proportion varies widely. Because the hydrophobic portions of membrane lipids resist mixing with water, they create barriers that separate the cell from its environment and divide the cell into internal compartments. Adipocytes are specialized cells that store large amounts of triglyceride. In a mature white adipocyte, one large lipid droplet can occupy nearly the entire cell. Stored triglycerides represent the body's major long-term energy reserve because fat provides more energy per gram than carbohydrates or proteins. Carbohydrates generally provide less of the cell's permanent structure, although carbohydrate groups attached to proteins and lipids are crucial components of cell membranes. Most human cells do not maintain large carbohydrate reserves. Carbohydrates average around 1% of the mass of many cells, but the amount may reach approximately 3% in skeletal muscle and 6% in the liver because these tissues store glycogen. Glucose is continuously available in extracellular fluid and can be transported into cells for energy production. Glycogen is an insoluble, branched polymer of glucose that functions as a rapidly accessible intracellular glucose reserve. Liver glycogen helps maintain blood glucose between meals, while skeletal muscle glycogen mainly supplies energy to the muscle itself. Carbohydrate chains attached to proteins form glycoproteins, while those attached to lipids form glycolipids. On the cell surface, these molecules contribute to cell recognition, adhesion, signaling, immune responses, tissue development, and fertilization. The nucleus, the cellular information center the cell, can now be viewed as having two major regions, the nucleus and the cytoplasm. The nucleus stores most of the cell's DNA, while the cytoplasm extends from the plasma membrane to the nuclear envelope and contains the organelles and cytosol. The nucleus is often described as the control center of a eukaryotic cell. This does not mean that it consciously directs the cell. Instead, it stores DNA-containing genes that encode RNAs and proteins. By regulating which genes are expressed, the nucleus influences cellular growth, maturation, metabolism, replication, specialization, and programmed death. The nucleus is usually the largest organelle and is commonly spherical or ovoid. Its appearance varies according to cell type and activity. The DNA inside it undergoes replication before cell division and transcription when genetic information is copied into RNA. Not every human cell has a nucleus. Mature red blood cells lose their nuclei during development, allowing more space for hemoglobin. Platelets are cytoplasmic fragments rather than complete nucleated cells. By contrast, most actively dividing eukaryotic cells require a nucleus because their DNA must be replicated and distributed to daughter cells. Nuclear envelope and nuclear pores. The nuclear envelope consists of two lipid bilayer membranes, an inner nuclear membrane and an outer nuclear membrane. The outer membrane is continuous with the rough endoplasmic reticulum. The space between the inner and outer nuclear membranes, called the perinuclear space, is continuous with the lumen of the endoplasmic reticulum. The envelope is interrupted by nuclear pore complexes. These are not simple holes. They are large, highly organized assemblies made from proteins called nucleoporins. Many nucleoporins are arranged with eightfold symmetry, forming cytoplasmic and nucleoplasmic rings around the pore. The pore contains a central selective transport channel. Phenylalanine glycine, or FG-repeat nucleoporins, form a selective molecular barrier. Small molecules can pass by passive diffusion, but large proteins and RNA molecules require regulated transport. Proteins meant to enter the nucleus usually contain a nuclear localization signal, while molecules leaving the nucleus may contain a nuclear export signal. Transport receptors and the RAN-GTP system provide direction and selectivity. On the nuclear side, filamentous nucleoporins form a nuclear basket. The nuclear lamina lies beneath the inner nuclear membrane. It is composed mainly of lamin proteins, which are intermediate filament proteins. The lamina mechanically supports the nucleus, helps organize chromatin, and participates in DNA replication and nuclear reassembly following mitosis. Mutations in lamins can produce diseases called laminopathies, including certain muscular dystrophies and Hutchinson-Gilford progeria syndrome. Chromatin, chromosomes, and the genome DNA inside the nucleus is associated with histone and non-histone proteins, forming chromatin. A long DNA molecule wraps around histone proteins to form nucleosomes. This packaging allows a very long DNA molecule to fit within the nucleus while still permitting controlled access for replication and transcription. In female somatic cells, one X chromosome is largely inactivated early in embryonic development. The inactive X becomes tightly condensed and can appear near the nuclear membrane as a bar body, also called sex chromatin. As a useful rule, the expected number of bar bodies is the number of X chromosomes minus 1. A typical Roman 20 cell therefore has one bar body, while an XY cell generally has none. X inactivation is not absolutely complete because some genes escape inactivation. The Fuelgen reaction is a histochemical method used to demonstrate DNA. It depends on a chemical reaction with deoxyribose and is considered DNA-specific under proper conditions. Chromatin is commonly described as heterochromatin or euchromatin. Heterochromatin is tightly packed and usually stains darkly. Because the DNA is relatively inaccessible to transcription machinery, genes in heterochromatin are generally inactive or transcriptionally repressed. Heterochromatin often appears near the nuclear periphery and around the nucleolus and tends to replicate later during S phase. Euchromatin is more loosely packed and stains more lightly. Its open structure makes DNA more accessible for transcription, so euchromatin is generally associated with active gene expression. Cells producing large amounts of protein often have pale, euchromatin-rich nuclei and prominent nucleoli. An important examination trap is to equate euchromatin with a complete absence of chromatin. Euchromatin is still DNA associated with proteins, it is simply less condensed. Another trap in the notes is the claim that heterochromatin constitutes 97–98% of the genome, while euchromatin constitutes only 2–3%. These are not reliable universal proportions and should not be memorized. As a cell prepares for mitosis or meiosis, chromatin becomes progressively condensed into visible chromosomes. Condensation reaches its maximum during metaphase, which is why metaphase chromosomes are commonly used for karyotyping. Before division, every chromosome must be replicated. The two identical copies are called sister chromatids and remain connected until they separate during cell division. Telomers are repetitive DNA protein structures at chromosome ends. They prevent chromosome ends from being mistaken for broken DNA and protect against degradation and end-to-end fusion. Telomeres shorten with repeated division in many somatic cells, whereas telomerase can maintain them in germ cells, some stem cells, and many malignant cells. Sex chromosomes determine chromosomal sex and carry additional genes. The X chromosome is large and contains many genes unrelated to sexual development. The Y chromosome is much smaller and carries fewer genes. One of its most clinically important regions contains the SRY gene, which initiates testicular differentiation during embryonic development. The genome is the complete genetic material of an organism. A normal human somatic cell contains 46 chromosomes arranged into 23 pairs. 22 pairs are autosomes, and one pair consists of sex chromosomes. One member of each pair is inherited from the mother and the other from the father. A typical chromosomal female has Roman 20 chromosomes, while a typical chromosomal male has XY chromosomes. Human gametes are haploid and therefore contain 23 chromosomes rather than 46. Nucleoplasm and nuclear matrix. The nucleoplasm is the gel-like internal environment of the nucleus. It suspends chromatin, nucleoli, enzymes, nucleotides, and regulatory proteins. It supports nuclear shape and allows materials to move between nuclear structures and nuclear pores. The term nuclear matrix refers to a proposed internal framework associated with chromatin organization, replication, transcription, RNA processing, steroid receptor interactions, heat shock responses, and viral activity. Its exact nature remains more complex and controversial than older descriptions suggest. It should not be imagined as a rigid skeleton filling the nucleus. Instead, the nucleus contains dynamic networks involving the nuclear lamina, chromatin-associated proteins, ribonucleoprotein complexes, and residual nucleolar structures. The nucleolus and ribosome production. The nucleolus is a dense, non-membrane-bound structure inside the nucleus. It stains basophilically because it contains large amounts of ribosomal RNA, or rRNA, together with proteins. It is prominent during interphase, but disassembles during mitosis and reforms afterward. Its major function is ribosome biogenesis. Ribosomal RNA is transcribed, processed, and combined with ribosomal proteins to form the small and large ribosomal subunits. These subunits then leave the nucleus through nuclear pores and assemble into functional ribosomes in the cytoplasm. Cells actively producing proteins usually have large, prominent nucleoli. In some highly active cells, the nucleolus can occupy a substantial portion of the nucleus. The pale staining fibrillar center contains ribosomal DNA and associated transcription machinery. Around it lies the dense fibrillar component, corresponding broadly to the notes pars fibrosa, where newly transcribed pre-rRNA begins to be processed. Proteins such as fibrillarin and nucleolin participate in rRNA transcription and processing. The granular component, corresponding to the pars granulosa, is where later processing and assembly of ribosomal subunits occur. A nucleolar matrix or supporting network helps organize these activities. The notes mention inactive DNA in the fibrillar center and active transcription in the pars fibrosa. A more precise understanding is that the boundaries between these nucleolar regions are functionally related. rRNA genes and transcription machinery are associated with the fibrillar centers. Transcription occurs near their borders, early processing occurs in the dense fibrillar component, and later ribosomal assembly occurs in the granular component. Chemically, the nucleus contains DNA, multiple forms of RNA, proteins, nucleotides, and the phosphate and nitrogenous base components required to build nucleic acids. DNA contains genes that determine the amino acid sequences of proteins and regulate when those proteins are produced. Through these products, nuclear genes control both cytoplasmic and nuclear activities and permit cellular reproduction. The cytoplasm and cytosol. The cytoplasm is the portion of the cell between the plasma membrane and nuclear envelope. It includes the cytosol, organelles, cytoskeletal structures, inclusions, and vesicles. The cytosol is the intracellular fluid remaining when membrane-bound organelles and other solid structures are conceptually removed. It is composed mainly of water containing dissolved ions, metabolites, proteins, and other molecules. Many metabolic reactions, including glycolysis, take place in the cytosol. Dispersed throughout the cytoplasm are ribosomes, organelles such as the endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, and peroxisomes. Inclusions such as glycogen granules, lipid droplets, pigments, and crystals, and membrane-bound secretory vesicles. Ribosomes and protein synthesis. Ribosomes translate the nucleotide language of messenger, RNA, into the amino acid language of proteins. They are made of rRNA and proteins and contain a small and a large subunit. During translation, the ribosome reads the messenger RNA in groups of 3 nucleotides called codons. Each codon specifies an amino acid or a stop signal. Transfer RNA, or tRNA, carries an amino acid and contains an anticodon complementary to the mRNA codon. Correct codon-anticodon pairing allows the proper amino acid to be added to the growing protein. The ribosome then catalyzes peptide bond formation between adjacent amino acids. The catalytic activity of the peptidyltransferase center is largely carried out by rRNA, making the ribosome a ribozyme. After a peptide bond forms, the ribosome moves to the next codon through translocation. Repeated cycles elongate the polypeptide until a stop codon terminates translation. Free ribosomes and roughy R-bound ribosomes are structurally identical. Their difference is determined by the destination of the protein being synthesized. Free ribosomes mainly produce proteins that remain in the cytosol or are imported into structures such as the nucleus, mitochondria, or peroxisomes. Ribosomes become attached to the rough ER when a signal peptide directs the growing protein into the secretory pathway. These ribosomes produce proteins destined for secretion, lysosomes, or cellular membranes. Membrane-bound organelles. Organelles are metabolically active, specialized cellular structures. Most are enclosed by membranes composed primarily of lipids and proteins. These membranes create compartments, allowing incompatible reactions to occur separately and allowing the cell to establish different concentrations of substances in different locations. The hydrophobic interior of a membrane restricts the movement of ions and most water-soluble molecules. Membrane proteins then provide selective pathways through which particular substances can cross. Endoplasmic reticulum. The endoplasmic reticulum, or ER, is an interconnected network of flattened sacs and tubules called cisternae. The fluid-filled space inside it is the ER lumen. It is chemically distinct from the surrounding cytosol. The rough endoplasmic reticulum, or RA, has ribosomes attached to its cytosolic surface. Proteins entering the secretory pathway are inserted into the ER membrane or translocated into its lumen as they are synthesized. Inside the RER, proteins begin folding, forming disulfide bonds, and undergoing initial glycosylation and quality control inspection. Misfolded proteins are usually retained and targeted for degradation. The smooth endoplasmic reticulum, or SER, lacks attached ribosomes. It synthesizes lipids, phospholipids, cholesterol, and steroid hormones. It also participates in drug detoxification, particularly in hepatocytes, and stores calcium in specialized forms such as the sarcoplasmic reticulum of muscle. Newly synthesized lipids become incorporated into the ER membrane. Small transport vesicles bud from transitional regions of the ER and carry proteins and lipids toward the Golgi apparatus. This trafficking also prevents uncontrolled expansion of the ER. The Golgi apparatus receives proteins and lipids from the ER. It modifies, sorts, and packages them according to destination. Proteins generally enter at the cis face, pass through medial cisternae, and leave from the trans face. Golgi modifications include further glycosylation, sulfation, phosphorylation, and proteolytic processing. The Golgi then sorts products into vesicles destined for the plasma membrane, secretory granules, or lysosomes. Mannose 6-phosphate is a particularly important Golgi-added targeting signal that directs many enzymes to lysosomes. Mitochondria are commonly called the powerhouses of the cell because they produce most ATP through oxidative phosphorylation. They are concentrated in cells and cellular regions with high energy demands. Cardiac muscle cells, renal tubular cells, and the ion-transporting regions of many cells contain numerous mitochondria. Mitochondria contain their own circular DNA, ribosomes, and replication machinery. Mitochondrial DNA is inherited overwhelmingly through the mother in standard clinical genetics. However, mitochondrial function still depends heavily on nuclear genes because most mitochondrial proteins are encoded by nuclear DNA and imported into the organelle. Mitochondria also regulate apoptosis. When a cell receives a strong intrinsic death signal, mitochondrial outer membrane permeabilization can release cytochrome C into the cytosol. Cytochrome C participates in apoptosome formation and caspase activation, eventually producing controlled cellular dismantling. Mitochondria are dynamic rather than stationary bean-shaped structures. They repeatedly undergo fusion, fission, movement, and turnover. These processes help distribute energy capacity, isolate damaged components, and maintain mitochondrial quality. Lysosomes are membrane-bound organelles containing acid hydrolases. These enzymes digest proteins, nucleic acids, polysaccharides, and lipids. Their activity is optimized in the acidic lysosomal lumen, which helps protect the cytoplasm because the enzymes are less active at normal cytosolic pH. Lysosomes digest material taken into the cell by endocytosis or phagocytosis and recycle damaged organelles through autophagy. This is why they are sometimes called the cell's garbage disposal or recycling system. The older term suicide sacs can be misleading. Lysosomes can contribute to cell injury under some circumstances, but apoptosis is primarily a tightly regulated signaling process involving caspases and often mitochondria. Lysosomes should therefore be remembered mainly as intracellular digestive and recycling organelles. A clinical connection is the lysosomal storage diseases. If one lysosomal enzyme is absent, its substrate accumulates inside cells. Examples include Tay-Sachs disease, Gaucher disease, and Pompe disease. Paroxysms resemble lysosomes morphologically, but have different enzymes and functions. They contain oxidases that generate hydrogen peroxide and catalase that converts hydrogen peroxide into water and oxygen. Paroxysms are especially important for the boxidation of very long-chain fatty acids, the metabolism of certain toxic molecules, and the synthesis of plasmalogens used in myelin. They grow and divide from pre-existing paroxysms, but also receive proteins and membrane components through coordinated cellular pathways. Paroxysomal dysfunction produces disorders such as Zellweger spectrum disorders and X-linked adrenoleukodystrophy. These diseases emphasize that paroxysms are particularly important in lipid metabolism and nervous system function. Cytoplasmic inclusions and secretory vesicles, unlike organelles, cytoplasmic inclusions are generally non-living accumulations that do not perform independent metabolic functions. They are usually not surrounded by membranes. They may represent stored nutrients, metabolic products, pigments, or crystals. Lipid droplets store triglycerides. They are abundant in adipocytes but can also appear in hepatocytes and other cells. Glycogen granules store glucose and are especially abundant in liver and skeletal muscle. On electron microscopy, glycogen can appear as clusters or rosettes and may be found near the smooth ER. Pigments include melanin, lipofuscin, and hemosiderin. Some cells may also contain crystalline inclusions. Secretory vesicles are different because they are membrane-bound and participate actively in transport. They commonly form through the Golgi pathway and carry proteins, hormones, enzymes, neurotransmitters, and other molecules to their destinations. When a secretory vesicle fuses with the plasma membrane and releases its contents, the process is called exocytosis. The cell membrane, the plasma membrane, forms the cell's boundary, but it is much more than a passive wall. It controls the movement of substances, receives signals, mediates adhesion and recognition, anchors the cytoskeleton, maintains electrochemical gradients, and communicates with the extracellular environment. The exact chemical composition varies among cell types. The notes give an approximate composition of 55% protein, 25% phospholipid, 13% cholesterol, 4% other lipids, and 3% carbohydrate. These values are useful as one example, but they are not universal. A myelin membrane is much richer in lipid, whereas the inner mitochondrial membrane is exceptionally protein-rich. Membrane lipids and amphipathic organization, the major membrane lipids are phospholipids, glycolipids, and cholesterol. These lipids are amphipathic, meaning that they contain both hydrophilic and hydrophobic regions. Phospholipids have polar, hydrophilic heads that interact with water and nonpolar, hydrophobic fatty acid tails that avoid water. In the bilayer, the heads face the extracellular fluid and cytosol, while the tails face inward toward one another. This arrangement forms a stable hydrophobic interior. The hydrophobic core makes the membrane relatively impermeable to ions and most large or strongly polar molecules, including sodium, potassium, glucose, and many proteins. Small nonpolar or lipid-soluble molecules, such as oxygen, carbon dioxide, and steroid hormones, cross more easily by dissolving in the bilayer. Water crosses to a limited degree through the lipid bilayer, but moves much more rapidly through aquaporin channels in many cells. Ethanol can cross membranes relatively easily, whereas urea crosses only to varying degrees depending on the membrane and its transporters. Membrane asymmetry, the plasma membrane is asymmetric. Its two leaflets do not have identical lipid, protein, or carbohydrate compositions. Carbohydrate chains on glycoproteins and glycolipids are found almost exclusively on the extracellular surface. Certain peripheral proteins are present only on the cytoplasmic side. Transmembrane proteins also have fixed orientations. A receptor's ligand-binding region may face outside the cell, while its signaling domain faces the cytoplasm. This orientation is essential for proteins such as the sodium-potassium at PACE. For every ATP hydrolyzed, the pump moves 3 sodium ions out of the cell and 2 potassium ions into the cell. It can establish these gradients only because its extracellular and cytoplasmic surfaces are oriented correctly. Phospholipids are also asymmetrically distributed. Phosphatidylcholine and sphingomyelin are enriched in the outer leaflet, while phosphatidylserine and phosphatidylphenolamine are enriched in the inner leaflet. When phosphatidylserine moves to the outer surface, it can signal apoptosis and mark the cell for phagocytic removal. Spontaneous movement of phospholipids from one leaflet to the other, called flip-flop, is slow because the polar head would need to cross the hydrophobic core. Enzymes regulate this process. Flipases and flopases move selected lipids in specific directions, while scramblases can rapidly reduce asymmetry. Cholesterol moves between leaflets more readily than phospholipids, so its distribution is more dynamic than the notes suggest. Regional asymmetry also produces specialized membrane domains. Examples include the apical brush border of intestinal cells, tight junctions, gap junctions, synapses, and lipid rafts. Each region contains proteins and lipids suited to a particular function. Membrane proteins, integral membrane proteins, are embedded within the lipid bilayer. Many span the entire membrane and are called transmembrane proteins. They function as channels, carriers, pumps, receptors, enzymes, adhesion molecules, and anchors. Channels form hydrophilic pores that allow selected substances to cross. Aquaporins facilitate water movement. Potassium channels selectively conduct potassium because their selectivity filters coordinate dehydrated potassium ions more effectively than other ions. Carrier proteins bind particular substances and change conformation to move them across the membrane. Carriers may mediate facilitated diffusion down an electrochemical gradient or active transport against a gradient. Therefore, it is not correct to say that all carriers transport substances against their gradients. The sodium-glucose cotransporter, for example, uses the sodium gradient to drive glucose uptake, while glut transporters move glucose by facilitated diffusion. Receptors bind extracellular signaling molecules and initiate intracellular responses. This is especially important for peptide hormones, which generally cannot cross the lipid bilayer. Insulin binds its membrane receptor and activates an intracellular signaling cascade rather than entering the cell to act directly. Peripheral membrane proteins attach to the surface of the membrane or to integral proteins, but do not pass through the hydrophobic core. They may function as enzymes, cytoskeletal anchors, or signaling components. The notes describe peripheral proteins as carrier proteins, but this is an exam trap. True membrane carriers that move substances across the bilayer are generally integral transmembrane proteins. Membrane carbohydrates and the glycocalyx membrane carbohydrates are usually attached to proteins or lipids, forming glycoproteins, proteoglycans, and glycolipids. Their carbohydrate chains project from the extracellular surface, creating a carbohydrate-rich coat called the glycocalyx. The glycocalyx protects the cell surface from chemical and mechanical damage. It also contributes to cell adhesion, receptor interactions, immune recognition, and tissue organization. Blood group antigens are clinically important examples of cell surface carbohydrates. These molecular labels help the immune system distinguish self from non-self, although immune recognition is not based on carbohydrates alone. The fluid mosaic model, Singer and Nicholson, proposed the fluid mosaic model in 1972. It describes the plasma membrane as a dynamic phospholipid bilayer containing a mosaic of proteins, cholesterol, and carbohydrates. Most plasma membranes are approximately 510 nanometers thick. The word fluid means that many lipids and proteins can move laterally within the plane of the membrane. The membrane is therefore flexible and can change shape. The word mosaic refers to the diverse components embedded in or attached to the bilayer. The membrane is not uniform. Different proteins and lipid domains have different functions. The common analogy is of proteins floating like islands in a lipid sea. This is helpful but incomplete because many proteins are anchored to the cytoskeleton, extracellular matrix, or other proteins, and therefore do not move freely. Membrane fluidity refers to how easily the lipids and proteins move within the bilayer. It is essential for transport, signaling, membrane fusion, endocytosis, exocytosis, and cell motility. Unsaturated fatty acids generally increase fluidity because their cis double bonds introduce bends or kinks. These kinks prevent phospholipid tails from packing tightly. Saturated fatty acids are straighter and pack more closely, tending to make the membrane less fluid. Shorter fatty acid chains also generally increase fluidity because they have weaker hydrophobic interactions. Temperature affects fluidity. At low temperatures, phospholipids move less and pack more closely, making membranes more rigid. At higher temperatures, molecular movement increases and the membrane becomes more fluid. Cholesterol acts as a fluidity buffer. At low temperatures, it prevents phospholipid tails from packing too tightly, preserving fluidity. At high temperatures, its rigid ring structure restrains excessive phospholipid movement and reduces fluidity. Cholesterol also reduces the membrane's permeability to many small water-soluble molecules. Fluidity allows membrane proteins to interact laterally, allows vesicles to bud during endocytosis, permits membranes to fuse during exocytosis, and supports shape changes during cell movement. A white blood cell, for example, must deform its membrane and cytoskeleton to leave a blood vessel and enter inflamed tissue. Antigenicity and functions of the cell membrane. A cell membrane is antigenic when its surface molecules can be recognized by the immune system and potentially provoke an immune response. Proteins, glycoproteins, and glycolipids may serve as antigens. ABO, blood group antigens, and major histocompatibility complex molecules are clinically important examples. Overall, the cell membrane maintains structural integrity, creates a selectively permeable boundary, regulates cell-to-cell interactions, recognizes foreign and altered cells, provides receptors for signaling, anchors transport systems, maintains electrical potential differences, and converts extracellular chemical or physical signals into intracellular events. That conversion is called signal transduction. Selective permeability does not mean that the membrane simply chooses substances consciously. It means that the lipid bilayer and membrane proteins have physical and chemical properties that allow some substances to cross more readily than others. Cytoskeleton. The cytoskeleton is a dynamic network of protein filaments and tubules. Its precursor proteins are synthesized on ribosomes and then polymerized into microfilaments, intermediate filaments, or microtubules. The cytoskeleton maintains cellular shape, organizes the cytoplasm, moves organelles, assists cell division, and allows cellular movement. Microfilaments are approximately 7 nanometers in diameter and are composed mainly of actin. They are especially abundant beneath the plasma membrane, where they form a supportive cortical network in the ectoplasm. Actin filaments participate in cell locomotion, changes in cell shape, attachment to the extracellular matrix, movement of the cell surface, cytokinesis, and developmental folding of epithelial sheets. During cell division, actin and myosin form a contractile ring that pinches one cell into two daughter cells. Myosin is a motor protein that interacts with actin. In skeletal and cardiac muscle, thin filaments contain actin, tropomyosin, and troponin. Calcium binds troponin, moves tropomyosin away from actin's myosin-binding sites, and allows contraction. This specific troponin arrangement should not be generalized to every cellular actin filament because many non-muscle actin filaments do not contain the same contractile regulatory complex. Microtubules are approximately 25 nanometers in diameter. They are hollow, relatively stiff tubes built from and tubulin dimers. Their polarity gives them a minus end and a plus end. Microtubules help maintain cellular organization, form the mitotic spindle, support cilia and flagella, and provide tracks for intracellular transport. Vesicles, granules, mitochondria, and other organelles can be moved along these tracks by motor proteins. Kinesins usually move cargo toward the microtubule's plus end, which is commonly directed toward the cell periphery. This is generally called anterograde transport. Cytoplasmic dynein usually moves cargo toward the minus end, commonly directed toward the centrosome or cell body. This is generally called retrograde transport. In a neuron, kinesin commonly carries vesicles and proteins from the cell body toward the axon terminal, while dynein carries recycled materials and signaling endosomes back toward the cell body. This distinction is highly testable, although some specialized kinesins move toward minus ends. Microtubule-associated proteins, or MAPs, bind to microtubules and influence their stability, spacing, organization, and interaction with cargo. They can prevent inappropriate depolymerization and help coordinate intracellular transport. During mitosis, microtubules and associated motor proteins position and separate chromosomes. Intermediate filaments are approximately 10 nanometers in diameter, placing them between microfilaments and microtubules. Their principal function is mechanical strength. They resist tensile stress and help maintain the three-dimensional structure of cells and tissues. Different cell types express different intermediate filament proteins. Keratins occur in epithelial cells and strengthen attachment to other cells and the basal lamina. They are clinically useful tumor markers because carcinomas commonly retain keratin expression. Desmin occurs in muscle cells. It links contractile structures and helps distribute mechanical force throughout the cell. Vimentin occurs mainly in mesenchymal cells, including fibroblasts, and helps support the nucleus and cellular architecture. Tumors of mesenchymal origin often express vimentin. Neurofilaments provide structural support to neurons, especially their axons. Their amount contributes to axonal diameter, which influences conduction velocity. Glial fibrillary acidic protein, or GFAP, is an intermediate filament protein strongly associated with astrocytes in the central nervous system and is commonly used as a histologic marker. The note's broad statement that glial filaments characterize non-neuronal cells throughout both the CNS and PNS should therefore be interpreted cautiously. A useful distinction is that microfilaments mainly support movement and contraction, microtubules support transport and chromosome movement, and intermediate filaments mainly provide tensile strength. In vivo, in vitro, and ex vivo studies, an in vivo study is conducted within a living organism. The phrase means within the living. Testing a medication in a mouse or conducting a clinical trial in human participants are in vivo approaches. These studies preserve the complex interactions among organs, metabolism, immune function, and circulation, but they may be more expensive and harder to control. An in vitro study is performed outside a living organism in an artificial, controlled environment. The phrase means within the glass. Examples include growing cells in culture, studying an enzyme in a test tube, or testing antibiotic susceptibility on bacteria in a laboratory dish. In vitro studies allow precise control of variables but cannot completely reproduce whole organism physiology. An ex vivo study uses cells, tissues, or organs removed from a living organism and studied outside the body while attempting to preserve aspects of their natural organization. Studying a freshly removed blood vessel's response to a drug is an ex vivo experiment. The important difference is that in vivo preserves the whole living organism. In vitro studies isolated biological material under artificial conditions, and ex vivo examines material freshly removed from an organism. A result observed in vitro does not automatically produce the same result in vivo because absorption, distribution, metabolism, immune responses, and tissue interactions can change the outcome. Mental summary: imagine moving from the smallest building material to the complete human being. Atoms form molecules, molecules build cells, cells form tissues, tissues build organs, and organs cooperate in organ systems. Inside each eukaryotic cell, the nucleus stores and regulates DNA, while the nucleolus produces ribosomal subunits. Ribosomes translate mRNA into proteins. The rough ER begins processing secreted and membrane proteins. The smooth ER produces lipids and handles detoxification and calcium, and the Golgi modifies and sorts cellular products. Mitochondria produce ATP and help initiate apoptosis. Lysosomes digest and recycle material, and peroxisomes perform specialized oxidative and lipid reactions. Surrounding everything is a fluid, asymmetric plasma membrane that controls transport, signaling, recognition, and electrical gradients. Beneath and throughout that membrane, actin filaments generate movement, microtubules provide transport tracks, and intermediate filaments provide mechanical strength. Every component depends on the others, which is the central message of the case. High-yield memory pearls remember the organizational sequence as chemical, cellular, tissue, organ, organ system, and organism. The four fundamental tissues are epithelial, connective, muscle, and nervous. Cell theory states that organisms are composed of cells, cells are the basic units of life, and cells come from pre-existing cells. Prokaryotes lack a membrane-bound nucleus and usually contain circular DNA in a nucleoid. Eukaryotes contain a true nucleus, linear chromosomes, and membrane-bound organelles. Human somatic cells have 46 chromosomes in 23 pairs, while gametes have 23 chromosomes. Dark, tightly packed, transcriptionally inactive chromatin is heterochromatin. Pale, loosely packed, transcriptionally active chromatin is euchromatin. The expected number of Barr bodies is the number of X chromosomes -1. The nucleolus is non-membranous, rich in rRNA and protein, and responsible for ribosomal subunit production. Free ribosomes generally make cytosolic proteins, while rough ER-bound ribosomes make secreted, lysosomal, and membrane proteins. The rough ER processes proteins, the smooth ER synthesizes lipids, detoxifies drugs, and stores calcium, and the Golgi modifies, sorts, and packages. Mitochondria produce ATP, contains circular maternally inherited DNA, and release cytochrome C during intrinsic apoptosis. Lysosomes contain acid hydrolases for intracellular digestion. Peroxisomes contain oxidases and catalase and metabolize very long-chain fatty acids. The plasma membrane follows the fluid mosaic model and is approximately 510 nm thick. Its phospholipids are amphipathic. Small non-polar molecules cross the lipid bilayer more easily, while ions and polar molecules usually require channels or carriers. Carbohydrates project from the extracellular surface as part of the glycocalyx. The sodium-potassium at PACE moves 3 sodium ions out and 2 potassium ions in per ATP. Unsaturated fatty acids increase fluidity by preventing tight packing. Cholesterol buffers fluidity. It preserves fluidity in the cold and limits excessive fluidity in the heat. Microfilaments are about 7 nm and are composed mainly of actin. Intermediate filaments are about 10 nm and provide tensile strength. Microtubules are about 25 nm, are composed of tubulin, and serve as intracellular tracts. Chitin generally moves toward the plus end in anterograde transport, while dynein generally moves toward the minus end in retrograde transport. Finally, do not confuse in vivo, in vitro, and ex vivo. In vivo means within the complete living organism. In vitro means under artificial laboratory conditions outside the organism. And ex vivo means studying freshly removed cells, tissues, or organs outside the body.